Hi there, Year 10. We are looking at these two pretty difficult questions for the topic B2, the distance learning questions that I set on Monday. And I'm just going to talk through so that you can green pen. If you didn't quite understand what each of the questions was getting at, hopefully you'll have a better idea. They're really quite recent questions, so very much in the style of what you're going to see in the real exam, okay? So question 1, oxygen is transported around the body by the blood. Blood leaving the human lung can carry about 250 milligrams of oxygen per litre. However, only 7 milligrams of oxygen will dissolve in 1 litre of water at body temperature. Suggest an explanation for the difference, and it's 2 marks, okay? So we know that oxygen travels around in the red blood cells, so that's already given us 1 mark to say that blood has red blood cells, or you could have said hemoglobin, which is found in those red blood cells. So that's our first mark, and our second mark is that hemoglobin can combine with the oxygen, so it can kind of cling onto it to form oxyhemoglobin. Now you don't actually have to write oxyhemoglobin, you could have got the second mark just by saying that the hemoglobin combines with the oxygen. Blood leaving the skeletal muscles during exercise may contain only 30 milligrams of oxygen per litre. So this is blood that's going away from muscles. So we're thinking it's dropping stuff off, it's dropping the oxygen off by diffusion for respiration. That's what's going on in my mind before I've even got to the question, okay? Because we're saying that the blood is going away from the muscles, and we know that that means it's going to drop off a lot of oxygen. It's saying at the top there that leaving the lungs, it's about 250 milligrams per litre, and we're now down to 30 milligrams a litre. Explain what causes the difference in oxygen concentration between the blood leaving the lungs and the blood leaving the skeletal muscles. Okay, so what we're saying is that at the lungs, the whole point of the blood circulating past the lungs is to collect oxygen, so it becomes oxygenated. Okay, first mark. Second mark is that it drops off that oxygen at the muscles. And why does it do that? Because of respiration, aerobic respiration. And why does that happen? Because the whole purpose of aerobic respiration is to release energy that can be used by those muscles in exercise. So 1 mark for saying that the blood becomes oxygenated at the lungs. Second mark for saying that it gets dropped off at the muscles. Third mark for saying why, for aerobic respiration. And 4th mark for saying that the point of aerobic respiration is to release energy for those muscles to use during exercise. Okay, hopefully that made sense. Quite a good question there, I think, because it really does get you thinking about kind of taking the information that we already know. We used a lot of our own knowledge there to apply to that situation. Bit of B2 there, also a little bit crossing over into B4 with the respiration there, but it's applying what we know to a new situation. We've got to think about everything that we've kind of locked in our minds from the learning that we've done, and how can we apply it to this situation. And that's exactly what we just did on that question. Okay, next question. So this is question 2, and there were only 2 questions. Amylase is an enzyme found in the human body. Amylase breaks down starch into sugar, okay, glucose. Where is amylase produced in the human body? Well, I know to take out the first 2 because no enzymes are made in the liver. It's actually the bile that's made in the liver. And I know it can't be the stomach because that's all about breaking down protein. So that's where the proteases are made, in particular pepsin. But I do know that when we chew our food, salivary glands release some amylase, so that's definitely a good one. And also the pancreas and the small intestine make all of the enzymes. So my process of elimination has taken me to the third option, salivary glands and pancreas. Okay, part B. Enzymes speed up chemical reactions. Explain how amylase breaks down starch. And this one is a 3 marker, okay? So we're saying explain how it breaks down starch. So first of all, we need to be very specific in referring to the active site. It's the fact that the starch, or you might have called it the substrate, fits into the active site of the enzyme, or you could be specific and say amylase as they have in the question. Okay, so 1 mark, starch fits into the active site of the amylase. 2nd mark, it's complementary, has a complementary shape. You could say about lock and key, you could say that it's the specific enzyme that's needed for that particular substrate, something that shows that you know that they match together. And the 3rd mark is the difficult one. Okay, what happens when that starch goes into the active site is that the bonds between those glucose molecules break. And I really emphasise this when I taught this in class. It's about the fact that those bonds that are holding all of those glucose molecules together are being chopped down into smaller units, and it's those bonds that are being broken by the enzyme. You might remember I had like a model of lots of glucose molecules velcroed together, and it goes into the enzyme and then that bond breaks. Another section of the starch that's now smaller will go into the enzyme and that will break. It's not some instant thing of it goes into the active site once and suddenly it's all broken down. The molecules get smaller and smaller until they become just single molecules of glucose. So they keep entering the active site, and a further section of the chain, the bonds are broken. Okay, moving on. 1 sugar in the body is glucose. Glucose is used for respiration. Give 1 other use for glucose in the body. Now don't get confused with SCARF, our uses of glucose in plants, okay? This bit we haven't really covered. We haven't talked about this word glycogen yet because it comes up in B5. Basically, where plants take their excess glucose and turn it into starch, we take our excess glucose and we store it as glycogen, okay, which is basically fat. So we will talk about that more in B5, so that might have been a tricky mark for you to access, okay. Because what we're saying here with this useful respiration, we can't just then list a load of things that we use the energy for. We can't say movement and thinking and all that kind of stuff, maintaining body temperature, because all of that comes from the respiration. So all we've got really is to talk about storage there. Okay, a student investigated the effect of temperature on the activity of human amylase. This is the method used. Put 2 centimetres cubed of 1% starch solution into a boiling tube. Put 2 centimetres cubed of amylase solution into a second boiling tube. Put both boiling tubes into a water bath at 20 degrees C. After 5 minutes, mix the amylase and starch together in 1 boiling tube. After 30 seconds, add a drop of the starch and amylase mixture to a drop of iodine solution in a well of a spotting tile. Okay, this feels familiar. This is a required practical. We've done this. What we did is we changed the pH of the amylase. Here we're changing the temperature. So taking that knowledge that we gained from the lessons where we looked at pH, and we're changing it to temperature. In actual fact, at our school we decided to do the temperature practical as well. So we have done changing temperature as well as changing pH, but the required practical does actually just talk about changing pH. So we put that into the spotting tile, repeat step 5 until the iodine solution no longer changes colour, so it stays that orange colour. Repeat 1 to 6 at 40 degrees C, 60 degrees C and 80 degrees C. Why did the student leave the starch and amylase solution in the water bath for 5 minutes in step 3? Because it's one of those things, it's like when people put a thermometer into a liquid and expect it to get to the temperature straight away. We've got to give it time to get to the temperature of our solution. So 5 minutes actually allows that liquid inside the boiling tube to acclimatise to the temperature of the water bath, which is 20 degrees C. So I've put to allow the starch and amylase to get to the same temperature as the water bath. Okay, we've got quite a tricky one here, a bit tricky to explain as well where the marks come from. The temperature of the human body is 37 degrees C. The diagram below shows the results of the investigation at 20 degrees C and at 80 degrees C. Complete the diagram to show the results you would expect at 40 and 60 degrees C. You should write a tick or a cross in each one of the spotting tile. Two things. One, you have to fill in every single section on the spotting tile. Two, you cannot do a tick, then a cross, and then go back to a tick. As soon as that tick becomes a cross, it needs to be a cross for the rest of the line. So we know that 40 degrees C is going to be a lot more effective than 20 degrees C because the particles have got more energy and they are going to be moving around more. There's going to be greater collisions. That links in with chemistry. So long as we have got two crosses at the end here, which is one more than at 20 degrees C, showing that it is a faster rate of reaction, we're good. So this one here could be a tick or a cross. Doesn't really matter. Here, likely to be a tick. It's unlikely that it would be so quick. If you put a cross there and you put crosses in all of them, then go for it. You can have that. But so long as you've got an extra cross here and potentially one here, that's fine. 60 degrees C, they could be ticks all the way along because you could identify there that it has denatured, just as you can see here on 80 degrees C, you can see that there was only starch present, it didn't break down. So you could go for all ticks under the assumption that the starch could not be broken down because the amylase is denatured. Or as you can see, I've got one cross at the end here where you could say, well, it might still work, but it might take a long time. Okay, so that's an optional one at the end there. Okay, moving on. Now I remember this question, and I particularly remember using this question as a mock paper question and people finding this bit quite difficult to get their heads around. So let's really try and get our head around it so that if something comes up like this again, then we're prepared, and that can happen quite often. There are different ways to investigate the breakdown of starch by amylase. Now, one of the main problems with how we have been doing it using the iodine solution is that people have got different perceptions of colour. So some people will say there's no starch left, the iodine solution isn't changing colour, and some people say, well, it's changing a little bit, I think there's still some starch left. So one other method is to measure the concentration of starch present in the solution every 30 seconds. Why is this method better than the method the student used? Okay, so they're actually measuring the concentration. Haven't told us how, they've just said that they can actually get a numerical value for measuring concentration. So that's the first mark. It's a quantitative result, therefore it's going to be more accurate. Okay, it's not going to have that subjective nature. And that's the second mark. It's less subjective because colour is a qualitative measure. Okay, so it's a quantitative measure rather than a qualitative measure. Okay, a colour is up for debate. Being given a number as measuring the concentration, you can't argue with that. You might have calibration issues, but if you've got that calibration issue, then it should apply to all your results, so they're still comparable. Okay, a calorimeter can be used to measure the concentration of starch present in the solution every 30 seconds. This is to do with putting light through the liquid and then getting a value as a result of how much light goes through. Okay, so they've briefly telling us here what a colorimeter is about. Okay, so the colorimeter measures the amount of light that cannot pass through a solution. This is known as absorbance because it's being absorbed by the liquid. Below shows a graph of absorbance against concentration of starch. So we've got concentration of starch at the bottom here, and then absorbance in that arbitrary units again. And we've come up with that one before. And we said arbitrary units is where the values are just relative to each other. So if I said to you, how happy are you on a scale of 1 to 10, and you said hopefully 10, then it's an arbitrary unit. It's just saying like a rough comparison. And then I might ask you another day and you might say 7. And so that's our arbitrary unit. It's not really a unit, it's just a comparison number. Okay, you can see I've drawn some lines in there. I'll come back to that in a second. So the absorbance of the solution at 40 degrees C was 0.56 arbitrary units after 30 seconds. What was the concentration of starch in the solution? So I've found 0.56, okay, look very, very carefully. So 0.5 is the halfway mark between, each little box is worth 0.02. So 0.56 is 3 little boxes up from halfway, and I've gone over to my line and I've come down, and then I read that off and it's 0.07, and the mark scheme doesn't allow any debate over that. It is just 0.07. okay, so you have to be quite accurate with your drawing there. The concentration of starch in the solution at 20 degrees C after 1 minute is different from the concentration at 40 degrees C after 1 minute. Explain why. So again, it links back to what we talked about earlier when we were looking at the spotting tile. It's about those collisions, the frequency of collisions based on the amount of kinetic energy that the particles have got. So at 20 degrees C compared to 40 degrees C, the particles have got less energy and they're moving more slowly. So therefore, the starch is being broken down more slowly. So 1 mark for saying at 20 degrees C, the starch is being broken down more slowly, or you could have said the opposite, at 40 degrees C, the starch is being broken down more quickly. Don't have 2 marks for that, it's just 1 mark for either way of saying it. And the reason is that the particles have less kinetic energy and fewer collisions at 20 degrees C, or you could have said have more kinetic energy and more collisions at 40 degrees C, and that gets our 2 marks. Now this one is a particularly challenging last mark question. Predict the absorbance for the solution at 80 degrees C after 30 seconds. Now the 30 seconds is pretty irrelevant because there's no time on our graph. But if we've got 80 degrees C, now we know from our spotting tile that 80 degrees C, nothing broke down. So if nothing's broken down, then all of the starch is remaining. And we can see here on our graph that this is the highest concentration of starch. This is all of the starch that they had in the solution at the beginning. They've not told us this, we've interpreted it from the graph. But if we're saying at 80 degrees C, it's all denatured, the enzyme can't work, then no starch will have been broken down. So we've got this concentration. So all I've done is I've taken a line across here using my ruler and I measure that off, and it's just below the midway point, so it's 1.08. So here's my answer, 1.08. No starch will have broken down as the amylase will denature at 80 degrees C. So we've got 1 mark for 1.08, 1 mark for the starch will not have been broken down, and 1 mark for it will have denatured by this temperature. Okay, I hope you found that useful. I hope you stuck with it and really kind of analysed those really difficult questions. Again, I'm going to say that second question was from a triple higher paper. So I know that I've set these questions for everyone. If you found it really difficult, don't panic. If you found it difficult but then understood it when I went through it, fantastic. And well done for just having a go. And we'll be moving on to B3 next week. Okay, so again, hope that was useful. Nice to really take some time to get into proper exam technique practice. Okay, have a good day. Thank you.